with Leslie Boshall in New York, where she began to actually focus on sort of plasticity aspects in coding in the bulb, or not the bulb, actually, this is my mammalian bias, in the antennal lobe, I should say, Freudian slip. After, I think, 3 years in New York, she returned to Berlin to work with Randolph Menzel, and then actually became a project leader at the Max Planck Institute for Chemical Ecology in Jena, where I think you should mostly switch to Drosophila as a sort of the animal of choice, and then began to focus on the higher centers of olfactory processing, like the lateral horn, and also on valence coding, how that is overall mediated. Currently, she is still at the Max Planck Institute, where she is a research group leader by now. But she's actually not only focusing on the higher brain centers, she's also working on aspects of the peripheral olfaction. I think recently she published a paper on the beetle that mediates Chagas disease, which makes me itch by just talking about it. So there's sort of a very broad aspects at looking at sort of invertebrate olfaction from the periphery to the center. And I think we'll hear more about most of this today. So welcome and looking forward to your talk. And over to you.
Thank you so much. Thank you so much, Johannes. Thanks for inviting me. Great pleasure to give a talk here at Monell and also to talk to you and other faculty people later on. So I will today, together with you, will dive into the olfactory circuits of Drosophila. I see there are also some flight people in the audience, which is nice. So I will share my screen. Here it is. Does it look good?
Yes, looks great. Thank you.
Perfect. So I'd like to talk about olfactory circuits and also especially plasticity in Drosophila, because recently we got very interested in plasticity of the circuitry. But so first of all, I would like to tell you about the aim of my group. So as Johannes said, I'm heading an independent research group here at the Max Planck Institute since several years, which is hosted by the Department of Bill Hanson. And my group has really the emphasis on Drosophila melanogaster. And we aim to understand how animals in general can take odor-guided decisions and to understand really the underlying olfactory circuits. For example, especially for flies or also other insects, for them it's really highly essential for their survival and reproduction to use their olfactory sense. For example, they need to find the right food sites. Here they would always go for the brownish yellow banana and never take those fresh unripe ones. And when they look for oviposition sites, they need to take decisions here when they fly around these apples. They, of course, should never lay their eggs on this moldy apple here because their offspring would die. So, and this moldy apple, it exposes a specific odor, which is geosmin, that we have studied together with Markus Densmir some years ago. So flies are completely repelled by this, and using their sense of smell, they will find the right suitable oviposition site so that their offspring can survive. And also for their communication, interspecific communication, or also interspecific communication, they use specific odors like sexual pheromones. And I would like to give you first some examples why Drosophila is really a premier model system for olfactory research. That's always important for vertebrate people to see why it's important also to work on insects. So Drosophila, the olfactory receptors, have been identified and sequenced in the late 90s by Leslie Fossel and John Carlson. And we know by now that they have about 60 odorant receptors on their olfactory organs. And Drosophila represents I would say, really one of the only organism where almost all of these odorant receptors have been functional characterized. So for each OR, we really know the ligand profile. It's very, very few that are still missing. So we do not have this information for any other animals. And then very important, of course, it's the olfactory system. Although it's very simple, it is analogous in structure and function to the vertebrate system, and all conclusions that we can draw from processing mechanism and coding mechanism, we can always really reflect this onto the vertebrate system. Now, our approach is always to link the neural circuits in the brain to the behavioral output to really understand what is the relevance of certain neurons, certain neuronal populations. And on the neural circuit side, as methods we use as one of the key methods. Now I pressed the wrong button and then it's always stopping. I need to stop the presentation. Now it's working. So we use two-photon functional imaging as a key technique in my group to study the representation of odors at different levels in the brain. We use different anatomical methods to visualize neurons. There we use a lot of photoactivatable GFP so that we can label single neurons or complete neuronal populations, and we reconstruct them using tracing and reconstruction softwares, and then represent these reconstructions in a standard brain so that we can compare neurons from different individuals. On the behavioral output side, we have many different behavioral assays in the labs. There I collaborate a lot with a group of Markus Knaden, who is heading the insect behavior group here at the Institute. So for just innate behavior, we use simple T-mazes, as shown here. It's just a simple two-choice assay. We use a flyborg. I introduce this later on. And right now we are establishing also a tethered flight setup, so that we can study odor-guided behavior while the fly is flying. And we also study reproductive behavior. We have courtship and oviposition assays to see which odors really would, for example, promote courtship or oviposition, which ones would inhibit it. And since we work with Drosophila, it's a genetic model organism, we of course use genetic tools to manipulate the system and really to understand what's the relevance of certain neurons population. There we use opto and neurogenetics, so optogenetics to artificially activate certain neurons, like for example, expressing crimson, or just different tools for silencing certain neurons. Now in the next few slides, I would like to walk you through the olfactory system of the fly so that you get familiar with it. So the nose of the flies have two olfactory organs. These are the antenna and the maxillary pulp. And when you would focus on these organs, you would see that these have these hair-like structures, which are the olfactory sensilla, and they exist in three different types: basiconics, siliconics, these small thick ones, and trichoids, these long-haired sensilla, which are mainly for pheromone reception. Now, when we go focus onto the brain, then the olfactory sensory neurons that are located in the olfactory sensilla, they then send their axons to the brain. And the first olfactory brain center, it's called the antennal lobe, the analogous structure to the olfactory bulb. And in flies, it just consists of 50 glomeruli. From there, output neurons in insects, they are called projection neurons, analogous to mitral cells. They then send their axons to higher brain centers, which are called the mushroom body. The mushroom body is a center for learning and memory in insects. And the lateral horn, the structure over here, which is probably an analogous structure to the cortical amygdala, and that's assumed to be the center for innate behavior. Now, when we focus on the antennal lobe, that's not just an input-output relay station, it's a complex network. And now my presentation stopped again. Sometimes stopping here. Now the local internet should come, but they don't come. So I need to unshare. I just stop share and then I just go out of the presentation. And now I play it again. Let's see. So I need to walk through this again. And I want it here, are the local interneurons. So the local interneurons, they form a complex network in the antennal lobe. It's a huge diversity of different neurons. There are about 200 in the fly, and they interconnect the different glomeruli and process the incoming olfactory information. Now the olfactory system has three essential tasks. It needs to encode an odor ligand, and This is doing with a set of odorant receptors, the repertoire of odorant receptors that are located and expressed in the olfactory organs. Then this information needs to be processed, and the first processing is really taking place in the antennal lobe. And then in higher brain centers, the odors need to be categorized into according to behavioral value, and then leading to a certain perception, and then enabling the animal to take a specific odor-guided decision. Now, during the last, I would say, 2 decades, many labs in many different species have shown that odors are coded by a combinatorial code. That's the case for the antennal lobe and also for the olfactory bulb. This means that each odor is inducing or activating a certain combination of glomeruli, like here, like benzaldehyde. So that's your Pfalz color code for activation. Red means strongly activated, blue is no activity. And benzaldehyde has this code in the antennal lobe in the fly. So it's this certain combination of glomeruli which really encodes the odor benzaldehyde. And a different odor has a different code. Now it's also from the glomerular side. Glomeruli are usually broadly tuned. So glomerulus can respond to either benzaldehyde or isoacetate in this case here. So it means the combinatorial code really means an odor activates several glomeruli, while a glomerulus can take part in the pattern of several odors. Now there's an exception for some labeled line odors. These are odors which activate a single odorant receptor class and therefore just a single glomerulus. And that's the case here, down here for this odor, cis-Wassenaar acetate. That's a male emitted sex pheromone in Drosophila, and has been shown that it activates only a single glomerulus. And we have a handful of odors that are also coded as labeled line codes, so that they only activate a single odorant receptor and therefore a single glomerulus. But the majority of odors is coded by a combinatorial code. Now, on top of this combinatorial code, as shown here on this side for different functional groups here, we could show some time ago that there is also a valence code. And that means that the behavioral value of an odor is also encoded in these patterns in a way that certain glomeruli, shown here in red, when those are activated, we could show that the odor is aversive. So aversive odors activate not all of them, but a few of them. And attractive odors activate the non-overlapping set of other glomeruli. So from the combinatorial code in the antennal lobe, we can predict for certain odors already the behavioral output. Now, in a natural environment, flies are, of course, not only exposed to monomolecular odors, they are also not only exposed to attractive or aversive odors, they could also be exposed to a conflicting sensory input, like in this case. It could be a food source where there's some mold on it. And now the fly needs to take the decision whether she oviposits here or whether this environment is too toxic and the offspring would die. So we therefore studied how does a fly really cope with the conflicting sensory input and how does a circuitry in the brain is processing such an information. So what we did 2 years ago is a study from Ahmed Mohamed, who did a PhD with me at this time. So we wanted to know how is a mixture of an attractive and a repellent odor coded in the brain. And first we studied the behavioral responses to such mixtures. So we used the fly walk. That's a behavioral assay that has been established by Markus Knaben in the lab. Here we study walking behavior in single flies. So each tube, there are 15 tubes, each tube contains one fly, and then the walking behavior of the flies is monitored. And then when we applied odors, for example, in green, ETA, it's acyl acetate. It's a highly attractive food odor for flies. When we apply acyl acetate at this concentration, flies start walking. So the gray bar here gives the odor application. That's the time, that's the walking speed. So flies start walking a lot because the odor is attractive for them. When we then apply benzaldehyde and aversive odor for flies, flies stop walking, or they don't even start walking. So they show a specific freezing behavior. When we mix now these two odors at this ratio, then we observe that the mixture is as attractive as the attractive odor. So it means that benzaldehyde has really no impact here for the mixture response. So we call this a mix plus, it's the attractive mixture. Now, when we lowered the concentration of acyl acetate by 10-fold, so 10 to minus 3, the odor is still attractive for flies, so they still walk a lot. Benzaldehyde, same concentration as here. Again, it's a freezing behavior. And when we mix now the two odors, at this certain ratio, we observe that the mixture is not as attractive anymore. It's still attractive, but less attractive than this one. So here the benzaldehyde has some impact. And now we wondered, these two mixtures, which induce a different behavioral output, so strong attraction, less attraction, they should be coded differently in the brain because they induce a different behavioral output. So we performed imaging of these odors. So we use two-photon functional imaging. We use specific transgenic lines where we expressed GCAM6F, so a calcium-sensitive reporter, under the control of just projection neurons, so the output neurons in the antennal lobe. And here's a two-photon functional images measurements. So you see the left, right antennal lobe. That's a response to benzaldehyde. Here you see the response to acyl acetate, the attractive odor, just as examples, and here the mixture of the two. Now, in order to be able to analyze those patterns, we need to really assign these neuronal responses to the glomeruli so that we can compare the pattern between each other and that we can compare the pattern between different flies. So what we did is we used the antennal lobe atlas that we developed some years ago. That's an in vivo atlas of the antennal lobe, where each glomerulus here is a specific color due to its input. So every glomerulus has a specific size, a specific position. It gets a genetically determined innervation of a certain class of sensory neurons that express a specific odor and receptors. So all neurons that express one or the same odor and receptor, they always converge in one glomerulus. And these glomeruli can just be named. They are the same from individual to individual, and then we can identify them and re-identify them in every fly and just assign our calcium responses to those glomeruli. This is what we did. And we did it now for the aversive odor, attractive odor, and the two different mixtures, the mix plus and the mix minus. And here you can see here are some names, arbitrary names of the glomeruli. And here you see the red circles are those glomeruli that respond mainly to benzaldehyde, to the aversive odor. And here in green, you see those glomeruli that respond to the attractive odor, acyl acetate. So the two odors activate non-overlapping sets of glomeruli, while the mixture then gives a mixture response to an overlapping set of the two odors. Now, to really analyze if there is a difference between the mix plus and the mix minus, we first represented those patterns using a principal component analysis just to see the representation. And here you see that benzaldehyde is clearly coded and represented differently from acyl acetate, both concentrations. The different dots give you single flies. And then when we looked on the mix plus, it's represented exactly in between. This is as you would expect it for mixture that's linearly coded. When we then looked on the mix minus, so the mixture that is less attractive, we observed that this is shifted towards the aversive odor. It's almost coded as the aversive odors. So these two mixtures are represented completely differently in the brain. And then we wondered, why do we have this shift here? And that probably also explains why mix minus is less attractive than mix plus. So we studied in detail the responses of the different glomeruli, and we focused on those glomeruli where we knew that they respond to either the aversive odor, shown here in red. So these are the two glomeruli that respond to benzaldehyde, the strongest activated one. And these are 4 glomeruli that respond to the acyl acetate. And when we compare the responses, here you see always the response to either benzaldehyde for the red ones, and in yellow the mixture, and here's the response to the acyl acetate, and here to the mixture. Then you see that the mixture response is always as high as the response to the stronger odor. So this you would expect for a linearly coded mixture. That's the mix plus. When we then looked on the mix minus, we observed that for those glomeruli that code the aversive odor, so we call them the aversive coding glomeruli, they also show the same mixture response as to the benzaldehyde, while those glomeruli that respond to the attractive odor, they show a very reduced mixture response. So they are decreased in their response, they are inhibited. And this explains why now the mix minus is coded almost like the benzaldehyde, like the aversive odor. Now we wondered, why do we have this reduction in the response? So there must be some inhibition going on, especially for this mixture, so some mixture inhibition. So we studied the underlying circuitry, the mechanism. We used some neurogenetics to silence specific glomeruli by silencing the odorant receptors. So we silence the input to the aversive coding glomeruli, this one that's now shown in gray because we silence it, and then we studied the response in the attractant coding glomeruli, and then this mixture inhibition was gone. So there we got already some hint that this aversive coding glomeruli probably induces or mediates the inhibition of those attractant coding glomeruli. Now, to really prove that, we used optogenetic activation. So we artificially activated the input to an aversive coding glomerulus by expressing crimson. So that's a light activated channel. So we just turned on the light, induced neuronal excitation in the aversive coding glomerulus. And then we observed that when we turned on the red light, that we could induce this inhibition in those glomeruli. So this shows that we have a clear inhibition from this glomerulus that is coding an aversive odor towards those glomeruli that are mediating the response to an attractive odor. We studied all the neurons involved in this and could identify a specific set of local interneurons, inhibitory local interneurons, which are GABAergic, that are responsible and that are mediating this crosstalk. And here it's just one example shown from these neurons. So this is a local interneuron that connects those glomeruli and that induces an inhibition from the DL1, so the aversive coding glomeruli, to the attractant coding glomeruli. So in the end, we came up with a model that we have a clear lateral inhibition taking place for odors that represent a conflicting input. So a repellent odor induces an inhibition, which is mediated by inhibitory local interneurons, to a glomerulus that is coding an attractive odor. And this inhibition then leads to a reduction of this input signal and then a reduced output signal. And in the end, we have a reduced attraction. So really the repellent odor is shutting down the input to the attractive odor. And then the fly is repelled suddenly by this attractive odor if a repellent odor is present. Now, odors do not always need to present a conflicting input. It could also be the case that two odors reinforce each other. And that's what we observed for food odors and pheromones. So when you observe flies in your kitchen, you will see that they always sit on food. So they do everything on food. They aggregate on food, they mate on food, they court on food, they oviposit on food. So we therefore wondered whether pheromone communication and food odor reception is somehow neuronal interacting. And indeed it is a case. So we could show also some years ago, we could show that really a food odor is interacting and boosting pheromone reception in flies. So male flies, they emit the sex pheromone cis-vacinyl acetate, which is coded like a labeled line. So when male flies release their sex pheromone, cis-vacinyl acetate, then the female flies get an activity in its pheromone responding glomerulus, and then it starts being interested in a courting male and then starts later on copulating with this male. Now, when we added food odor and we used vinegar, because vinegar flies really love vinegar, it's one of their food sources. When we added vinegar to the sex pheromone that's emitted by a male fly, then we observed that in the virgin female brain, the response in the pheromone encoding glomerulus was strongly increased. So it was a synergistic mixture response. And when we then observed the courtship in these flies, we observed that the female flies courted or copulated much earlier with a male fly when food was around. Also here we dissected the whole circuitry, and I just show you the result in the end. the circuitry showed us that there is a mixture synergism taking place in a way that those glomeruli that mediate or that respond to the food odor, they boost the activity of the sex pheromone responding glomerulus mediated by excitatory local interneurons. And in this case, the sex pheromone responding glomerulus is much stronger activated, has a stronger output, and then leads to an enhanced attraction. So here we have a case for reinforcing mixtures. This result, or when we published this study, that was pretty well recognized by the press, who then published that female, or hypothesized that female virgin fruit flies are more likely to have sex if they smell food. And they hypothesized that maybe something similar is happening in humans as well. So we have these two circuits now that I just showed to you. So conflicting input is mediated by lateral inhibition in a way that a repellent odor is suppressing the input to attractive odor and therefore then reducing the attraction to a food source where, for example, mold is around. While on the other way, we can have reinforcing mixtures in a way that a food odor is boosting the perception or is boosting the reception of the sex pheromone, and that leads then to an enhanced attraction. One is done by lateral inhibition, the other one is done by lateral synergism, and right now we are studying whether this is really a general neuronal mechanism for conflicting or for reinforcing mixture interactions. Now, I would like to come through some very recent studies and also still ongoing studies, and that's plasticity. So we understand more and more about the neuronal circuits in the brain, but now we are wondering, how plastic is this? What happens now when for example, the behavioral value of an odor changes? And what happens when a fly is raised to completely different environmental conditions? Can they adapt to it? So in one study, which is still ongoing, what we are doing is we take flies and we raise them in a completely different odor background. So we raise them in a background of geosmin. And geosmin is the odor of mold, and flies are strongly repelled by it. They would never oviposit there, and they would never feed on it because they know their offspring would die if they would really place their eggs in a moldy environment. Then some years ago, Markus Densme here, while he was here in the department, he could show that geosmin is encoded like a labeled line. It's one specific receptor, or 56A, that's responding to geosmin, which projects to the DA2 glomerulus. And then we also studied the circuit in the mushroom body and in the lateral horn. Now we wondered what happens now when flies are raised on a geosmin environment for several days. Does the underlying circuitry change, and can they get used to geosmin? Because the odor geosmin itself, it's not toxic. It just indicates a toxic environment, but the odor itself, it doesn't harm the flies. So what we did is we took flies, we put them in bottles with food and added the odor geosmin for 4 days. And then we started the circuitry. So we started the DA2, which is the geosmin responding glomerulus. We studied the branches in the antennal lobes, the branches in the mushroom body, the branches in the lateral horn, and so on. And we could show, just briefly summarizing it here, that in the mushroom body lateral horn, we didn't see any changes, which was extremely surprising, but we saw strong changes in the antennal lobe. So the glomerulus was increased in volume. That could be shown before. I could show it also for the CO2 circuitry while I was with Leslie Fossel. But now what we could show is the reason for this volume increase, and that could be found really in stronger arborizations of local interneurons and of projection neurons. So we see that's a lot of restructuring taking place in the antennal lobe. When we look at the behavioral level, we observed that flies, they can really get used to geosmin. So they don't like it, but they don't avoid it anymore. So here's the innate preference. Normally, these are flies that have been raised in a control environment, so with mineral oil, with a solvent, they are repelled by geosmin. But when they have been raised in a geosmin environment, then geosmin is suddenly neutral for them. And when we study oviposition, we see exactly the same. So normally flies are repelled by geosmin, while when they have been raised on geosmin, it's neutral. So they would also deposit the eggs in the geosmin environment. And currently, that's why I said the study is still ongoing. Currently, we are studying the functional level, so the pre and post synapses of the different neurons involved, to see where does a change really take place that would explain the change at the behavioral level. And now I would like to introduce to you another plasticity study that we are doing currently. So coming back to the valence map that I showed in the beginning. So in the internal lobe, as I said, we have a combinatorial code of odors, but we also have a valence code, meaning that there are glomeruli that code aversion and there are glomeruli that code attraction. Some years ago, we could show that this valence code is even further pronounced at the next level, which is the lateral horn, so the analogous structure to the cortical amygdala. And at the level of the lateral horn, we think that only or mostly behavioral value is encoded and that odor specificity is mainly lost. And we could show that the lateral horn consists of specific odor response domains, which just reflect behavioral value. So there's one big domain which codes attraction. So all odors that attractive would activate this domain. There's one domain which codes odor aversion. And we also identified one domain which is coding odor intensity. So when an odor is given in a high concentration, this domain was strongly activated. Now we wondered what happens now when we change the value of an odor, when we just switch the balance from attraction to aversion, then the code in the lateral horn should change accordingly. And that is what we are studying currently. So how do you change the balance of an odor? So we use aversive associative conditioning to change balances. So what we do, we use it's a T-maze assay. It's a so-called Tully machine. It has been established by Tim Tully many years ago, this learning assay, and it's used by most of the learning labs in the world. So it's a T-maze assay. So first you put flies in a tube with a copper grid. You give them, apply an odor, and an electric shock. So you pair an odor with a negative reinforcer. That's why you need this copper grid, so to really apply an electric shock. And afterwards, the flies are released down here with this little elevator, and then the flies can choose whether they go to odor A, that has been paired with a shock, or whether they go to odor B, which is a control odor. And we did it now with attractive odors. So we used attractive food odors. Odor A paired with a shock, then a control odor, odor B, and then we tested. So we use two odors, acyl butyrate and acetine acetate, which are both attractive and represent food. We use them at a concentration where they will equally attractive. So flies didn't really make a choice. So they usually distribute equally between the arms of these two odors. Now, when we trained acetone acetate with a shock, so when we made acetone acetate aversive, flies then were repelled by this odor and went into the arm for the ETB, so the acyl butyrate. When we switched it around and we paired acyl butyrate with a shock, we got exactly the same. So acyl butyrate is now aversive for flies, and flies prefer the acetone acetate. Now we were wondering, how does the code in the brain change accordingly, since we changed now the balances of these two odors? So what we did next is we focused on the projection neurons. So I showed to you first, so here's the antennal lobe. There are projection neurons that collect the information from the antennal lobe and convey to the higher centers. And there are two different types of projection neurons in the flies. One type, it's called the Uniglomolar projection neurons. They innervate single glomeruli, and they innervate the mushroom body calyx and the lateral horn. And there's a second population of projection neurons. They are multiglomerular, which means they innervate several glomeruli. They bypass, interestingly, the mushroom body, and they go directly to the lateral horn. We studied these neurons some time ago and could show that they are really coding, they are responsible for the odor response domains in the lateral horn that I showed to you, which really reflect the behavioral value, the odor valence. And we could also show that these neurons, that they mediate odor attraction. Now we wondered, since they mediate odor attraction, these neurons, they should change. They should be modulated when we change the balance of an odor. So what we did is we performed functional imaging of these multiglomerular projection neurons by using a specific line that specifically labels those neurons, and we expressed GCAM6F, so the calcium sensor just under control of these neurons. So now we did the training under the functional imaging setup and paired an odor with a shock while we applied the second odor as a control odor without any training. And then we did imaging in the lateral horn before and after training. So when we paired acetoin acetate with a shock, before we got a strong response in the lateral horn, after training, after this odor has shifted to aversion, we observed that the odor response was strongly reduced. As you can see it here by the time traces, and here you see the response of several individuals. So we have a decrease, and that's in the attraction domain of the lateral horn. When we then analyzed the control odor as albuterate, which was not paired with a shock, we got no change. So the response was the same before and after training. Now, when we shifted this around, so now we paired acylbutyrate with a shock, we observed exactly the same. So the response to the attractive odor was strongly reduced after training, while the control odor was represented exactly the same. Now we wondered, so we see a reduction in the attraction domain, which makes sense because the odor is not attractive anymore. Now we wondered what happens to the other two domains in the lateral horn. And interestingly there, we didn't see any change. So here in this case, acetoin acetate was paired with a shock, which you see the same response before and after training, and the same in the aversive domain. So these seem not to be modulated by learning. Then we wondered, maybe it's just the shocks, the electric shocks that's inducing this response decrease, or it's the long-term odor application. So what we did next is we used an unpaired training protocol, meaning that we uncoupled the shock from the odor application. So we just presented the shock, and later on we presented the odor. And when we did this unpaired training paradigm, we did not see any change. So this shows that this reduction in the response for the attraction domain is really due to the association of the electric shock with the odor. Now, we wondered, so these neurons, they are reduced in their activity, and these multiglomerular projection neurons, they are inhibitory, they are GABAergic, and they inhibit their postsynaptic partner neurons, which are third order neurons that innervate here the lateral horn and then send their axons to the next brain area in the fly, which is called the VLP, the ventrolateral protocerebrum. Now we wondered that these neurons, they should be now affected or modulated accordingly, since these neurons show a decrease in response. These neurons, since these neurons are inhibitory, these neurons should have an increase in their response. So what we did is we performed functional imaging of just these third order neurons using a specific line, expressing again GCAM, and then doing the training under the imaging setup. What we observed is indeed we have an increase in the response. So when we pair acetoin acetate with an electric shock and then measure pre and post, so before and after training, we see that these neurons, they are increased in their response. While for the control odors, acylbutyrate or benzaldehyde, which is an aversive odor, we didn't see any change. Yeah, so again, the association between the shock and the odor leads then to modulation also of these third order neurons. Then we wondered, so since a punishment leads to reduction in these neurons, which code attraction, what happens now if we make an odor even more attractive? So what we use is appetitive learning. So we applied a positive reinforcer while we applied the odors. And what we use is a sugar reward. When we applied the odor, we presented a sugar reward to the fly, and then we tested the flies afterwards. And interestingly, those neurons are really bimodally modulated, showing that those neurons after training can be increased in their response when an odor becomes even more attractive, while the control odor, which was presented without any reinforcer, is unchanged. Next, then we wondered, so since these neurons, they are bimodally modulated, they decrease after punishments or aversive conditioning, they increase their response after appetitive conditioning. And we know from our previous study that these neurons, and they code or mediate attraction. Now we wondered whether just the activity of these neurons can induce a memory, an attractive memory. And to study this, we used a genetic tool to artificially activate these neurons. We used the thermosensitive cation channel called TRPA1. When we increase the temperature, this channel opens and we can excite the neurons. What we did is, now it stopped again. I need to, and then go back. Sorry, always stops in between. Now it goes. So what we did now is we applied an odor, but we did not apply any reinforcer. So no sugar, no shock, but we just increased the temperature and therefore selectively activated those multiglomerular projection neurons. And when we did this, when we paired an odor with an activation of these multiglomerular projection neurons, the fly learned. So the fly learned that the odor that was presented with the activation of those neurons is attractive, and they went to the odor, while the parental controls, where we could not induce this activation, showed a neutral response. And when we did the temperature controls, and when we just applied the odor without any temperature increase, we didn't see any learning. So this shows that these multiglomerular projection neurons, that they are sufficient to induce an appetitive memory trace. Now, last slide to this part just gives you a model, what we think, what's going on right now. So those neurons down here, multiglomerular projection neurons, when we apply aversive olfactory conditioning, those neurons are reduced in their response. When we apply appetitive olfactory conditioning, those neurons are increased in their response. Their postsynaptic partner neurons are modulated accordingly. And by doing a series of other experiments, which I didn't have time to show to you now, we could show that glutamate as well as serotonin are involved in this modulation. So this shows that an innate circuit that is really mediating odor attraction can be modulated by olfactory learning. Okay, just very last is an outlook what we are currently doing. Right now we're trying to measure neuronal activity in behaving flies, and we have set up a tethered fly setup. So flies are just here fixed on a little needle just on their thorax so that they can be freely movable, and then we analyze and monitor their wing frequency and the amplitude of the wing so that we can see whether the fly flies faster or whether they turn towards or away from the odor. And currently we set this up at the two-photon microscope to image then the responses in the lateral horn to several odors, and then to see whether really we can correlate the flight behavior towards the odor, away from the odor, with the responses in the different domains in the lateral horn. That's currently ongoing. Last slide, I would like to summarize my talk briefly, the main take-home messages, what I've shown to you. So I showed to you that odors are coded by a combinatorial pattern of glomeruli in the antennal lobe. That's the same case, of course, in the olfactory bulb. And interestingly, at the next level, odors are rather than categorized according to the behavioral value, which we could show for the lateral horn. I showed to you with the two mixture studies that the conflicting sensory input is encoded by lateral inhibition, while the reinforcing sensory input, which means, for example, odor, which means a food odor and a pheromone, is encoded or mediated by synergistic interactions. And then both is modulating then or mediating then the behavioral output. And last, to the learning story, I showed you that these odor representations that we observe, they can be modulated by associative learning. Last slide, most important slide, it's my group in Jena. Here are my PhD students, so the learning studies from Florencia Capitella. Ahmed Mohamed worked on the conflicting sensory inputs, the mixture inhibition. He's now doing a postdoc with Charles Zucker. Benjamin Fabian is working on the geosmin exposure in flies. I didn't have time to talk about this project. Devasena, we work also on multimodal integration, and Devasena is working on how does a visual and olfactory input integrated in the brain, at what level, and can flies learn to associate a visual and an olfactory input. and Pramit and Soma have just started. Then Veit Grabe works on the tethered fly setup. Sudeshna works, is postdoc with me. These are my postdocs here. Sudeshna works on all the synergistic interactions. Anna De Petris-Chaumont is working on doing odor coding in different fly species. So we have generated also GCAMP constructs for other species that are non-Melanogaster. With Ching Zong, we work on locust olfaction. So we have just generated the first locust expressed in GCAM because locusts have a completely different architecture than any other insect. And we wonder how our odors represent at this level. And Karen also posted with me that we work on individuality. And we see, as probably all of you observe, there's a lot of variability taking place. And we are wondering whether this variability is real variability in behavior or whether individuality. And Silke Trautheim is technician with me in taking care of all the flies and dissections. And she's, extremely valuable for all projects that we are doing. Here are my collaborative partners. So I do a lot of projects with Markus Knaden together. I'm very grateful to Bill Hansson to host my research group in his department. With Johannes Felsenberg, we are doing the learning project together, also with Fernando Locatelli and Martin Karpenbach. And last but not least, the funding agency. And thank you very much for your attention.
Great. Thank you very much, Silke. I think that was fantastic. And I'm always in awe about the, genetic and imaging and recording possibilities that there are in the fly and what great science you can do with it. It always makes me feel like in the mouse system, we are still decades behind, somewhere in the Stone Age. If you have any questions, you can either put them in the chat, or you can unmute yourself and then ask them directly. So I can start things off. So you sort of talked about the sort of the aversive and the attractive parts of the antennal lobe, and then you sort of touched on how they feed forward into the lateral horn. Is that only true for the attractive side of things, or is there, looking at the multiple PNs, is there a similar system for the inhibitory, for the aversive side?
That's a very good question. So for the multiglomerular projection neurons, we just see really attraction. For the other projection neurons that I briefly introduced, the uniglomerular projection neurons, there we see, so these we studied also now in more detail in the lateral horn, and there we see both attraction and aversion. But it's not that one neuron type is only really taking care of just aversive odors. But I would say, so the uniglomerular, the other population of projection neurons, they have both, they encode both valences, attraction as well as aversion. But when we then study neurons in the lateral horn, there we really find a complete split. So there we find neurons that are only responding to aversive odors, or we find neurons that are only responding to attractive odors. So it seems to be that the split for odor valence is really taking place at third order neuron level.
Okay, so it gets a little bit primed at the periphery, but then sort of the final neuronal computation is actually then made at the third stage. Yeah. So when you looked at the attractive versus if you look at the mixture interactions, so it always seemed like the aversive one is dominating via the inhibition. Is it also the other way around, or is this sort of a one-way system? So which of the two, is the attraction or the inhibition more important?
Right. We also wondered about this and then tried to challenge the system to really change the ratios in completely the other direction. And we never found it in the other direction for this. And we had several odor combinations, and we only could see always that the repellent odors suppress attractive odors, never the other way around. And we also thought about it, why do we have it only in one direction? And then we just thought maybe it's ecological relevance at a repellent odor that's toxic. So it's more important to suppress it than really to override a repulsive odor. Maybe it's more important to override the attraction, because then it's maybe a dangerous environment for the insect and they really should not go there. So, but really, it's really directed only in one direction, as far as we see it for those odor combinations that we have tested, and as far as we have studied the circuitry.
Yeah, that's interesting. For the geomessine, if I remember correctly.
The geosmin, right. Geosmin is the same. It also kind of overrides in behavior, at least. So you can use very attractive odors. When you add geosmin, it overrides always the attraction.
Yeah, but you can
also if the attractive odors in a high concentration, you can always override it.
Yeah, but you can also make the fly used to it. So that's of interest.
You can make the fly used to it, and then you can really think about if this is a useful mechanism or not, that they get used to it, and then suddenly oviposits there, because if it would be the real environment, it's again moldy and the offspring would die, right? This we also discussed a lot, right? Why is this plastic? It's interesting that it's plastic, but is this really useful that it's plastic? But but it just shows that really hardwired circuits are also plastic. We did the same with pheromones. So we did aversive conditioning with pheromones. Usually pheromones are attractive, right? Because they also tell the fly that there are other flies. So we did aversive conditioning with pheromones to see, can they learn that this is something bad? And interestingly, they just learn it's neutral, but they would never learn that's really bad. So this seems to be pretty hardwired, and not that plastic.
Yeah. All right. Any other questions from the audience? Do we have any? Nothing in the chat.
Yeah, so I think you're making a strong claim here that each glomerulus basically has a fixed valence. Is that correct? That you feel
no. Okay. I don't say it for each glomerulus. I just say it for a really subgroup. A few glomeruli, we could show that they have a valence, but the majority, I would say it's rather neutral. So it's just a subgroup where we could show that they really correlate with activation of two attractive odors or activation of aversive odors.
I see. And if you take a bunch of novel odors and you just look at imaging, how well can you predict attraction or repulsion, or do you have a sense of how well it works?
Yes, okay. We haven't done it, but that's of course right. It's interesting to do, but we haven't. But when you look in the lateral horn, where I said there we have even a more pronounced segregation. When you there look on certain projection neurons and you see those ones that go to the aversive domain, then we could later on, then we already predicted this glomerulus should be aversive, because it really goes to the aversive area, and then we could really show that it's indeed, so really the segregation in the lateral horn, it's indeed correlated very well with the behavioral output, so that we could show it for some new odors. But I'm not claiming that all glomeruli have a certain valence code. That's not the case.
Okay, I was just checking. It sounded controversial. I like it.
And due to valence, there are probably other parameters which, of course, which represent behavioral values, which also play a role, which we haven't studied yet.
All right. Any other questions? Doesn't seem to be. So thank you very much. Yeah, it was great to learn what is happening in the fly and in the brain. And yeah, thanks again. Yeah,
thanks, Johannes,
for staying up and giving a talk.
Thank you so much.
Okay. Thank you so much. See you soon. Bye.
Bye.